I want to start today by talking a little bit about the innate immune system. Some of these mechanisms, all of the mechanisms here use the bacterial toolkit, the molecular toolkit from bacteria and prokaryotes that was, bacteria and eukaryotic single cells that was inherited by animals. And so as you were settling in, I hope you were looking through these and trying to explain them to yourself just from the sketch. I hope you'd be able to sketch this for yourself or close your eyes and be able to picture it, get through all of these step by step. So what's one? Skin, just a physical barrier. And two, okay, going to trap stuff and can be shed. Three, acid. Four, okay, digestive enzymes. The digestive enzymes you're using to break down food also serve perfectly well to kill invaders. What about five? What do those little circles represent? Okay, peptide antibiotics, those little circles represent amino acids. So it's a peptide. I think I drew something like 9 or 10 of them in here, some particular shape. And what's it doing? In this case, I've shown one that will bind to bacterial DNA, some segment of bacterial DNA at an essential gene, and that'll kill the cell. Equally, it can bind to some surface receptor. These peptide antibiotics can bind to something on the surface of the cell that will prevent some essential function. Or they can get into a bacterial cell and bind to an enzyme that's essential and kill it. Okay? So peptide antibiotics, common across all animals. What about 6? What am I representing here? Okay, complement system, that's a hole in a cell. We've just put a little donut with a big central pore into a bacterial cell, and that pore cell is leaking to death. Okay, that's the complement system. Your textbook has actually a really nice figure showing the complement system. It is so complex, it's definitely not worth your time to memorize how it works. It's a multiple protein complex, so multiple genes. I forget what the exact number is. It's on the order of a dozen or 20 genes. So all these different proteins that do more than one thing, but one of the major functions is make these little donuts, complex of proteins that will self-insert into a bacterial cell, cause it to leak to death. Okay, so that's the complement system. Does other things as well. What about 7? What am I showing in 7? Okay, interferons. So get an infected cell, that's our little one that's dying, the little X's and the unhappy smile, or maybe that's DNA. Anyway, that little cell is infected with viruses, it's doomed, but it can tell its neighbors, hey, I'm infected, protect yourselves. Okay? So what it's doing is releasing interferons, which will ramp up antiviral defenses in neighboring cells. Okay? And so the neighboring cells then may be protected against those viruses. What about number 8? Okay, agglutinins, not the best of names, but sticky. Okay, agglutinins, non-specific sticky molecules. If a cell or cells secrete proteins, or proteins that are maybe glycoproteins, so with sugar chains, that happen to stick to something on a bacterium, but not to anything else in the body. If you secrete these things, and if they're sticky at both ends, maybe it's a dimer that is attached in the middle, so it's sticky at both ends, then at one end it's going to stick to a bacterium, and as soon as another bacterium gets close to it, it's going to stick to that, and now these two bacteria are stuck together. You can imagine yourself, it may not seem like a big deal. Yeah, so what? You stick two bacteria together. Just to imagine yourself stuck to the person in the seat next to you for the rest of your life. Glued, super glued. And then imagine that as soon as you bump into someone else, the 3 of you are super glued to each other. And as soon as you bump into anyone else, all 4 of you, how easy, especially for the ones in the middle, how easy is it going to be to eat or do anything else? Okay, it's actually a pretty powerful defense. Okay, so non-specific agglutinins. Where'd number 9 go? Okay, what's number 9? Okay, gut flora. So competing gut flora. Bacteria that are not harmful, that are competing with whatever else is in there, with invaders. Number 10, where'd number 10 go? Number 10, what's number 10? Okay, phagocytic cells, a variety. For us, macrophages are a major category, but phagocytic cells that are just wandering around, they have their own independent little life. They're like your own little pool of amoebae, wandering around, and every time they bind to something that is on bacteria or viruses, but never part of you, they engulf it and digest it. Okay. What's 11? Okay, so that's a damaged cell secreting cytokine, and it's doing two things. What's it doing? It's attracting macrophages and causing inflammatory reaction, causing swelling. So I'm showing those little triangles attracting macrophages at the top, and at the bottom, it's doing things like opening up precapillary sphincters, causing vasodilation of arteries. So if the arteries get bigger, more blood comes in. If the precapillary sphincters open up, blood goes through those capillaries. It also tends to make the capillaries themselves loosen their attachments. So the capillaries get even leakier. So what happens? Tissue swelling, which by itself seems like pointless. Why just swell tissue? But if you want room for immune system cells to get in there, then you need to provide space for those immune system cells in between everything else, right? There's not a lot of space between cells normally, but if you allow fluid in, then amoeboid cells can creep in between those cells and get toward an attack, whatever's there. Okay, so these are great defenses. I want you thinking some about trade-offs. So would you get fewer infections if you had thicker skin? Skin infections. Sure you would. You wouldn't be able to be cut as well. If you had skin like an elephant or a rhinoceros, you can take a knife to a rhinoceros hide and hardly do a thing. It is like the hardest leather you have ever encountered. Okay? It is tough stuff. Sure you could. Is there a cost? Yeah. If you've got rhinoceros skin, guess how flexible that stuff is? Not very. Okay, so in there's a cost. How good do you think a rhinoceros is at detecting a tick crawling across the surface of its skin? Not as good as you are. Okay, so there's a cost. You could produce more mucus. Is there a cost to that? The mucus itself, the molecules itself, right? More acid in the stomach, you'd kill more things. Is there a cost? Okay, your cells are not going to last as long. Okay, same thing with more enzymes. You could produce more intense, more enzymes, making it a rougher environment, but it's also more hostile. And of course, there's some of the resource costs. What about peptide antibiotics? Any cost to them? Certainly the resources. Okay? And they're probably slower to evolve. And of course, that's an evolutionary arms race. The bacteria that are normal pathogens of a particular species, there's strong selection for them to favor alleles that make those either the peptide antibiotics to either make them not bind to a particular receptor, so just change the shape of the receptor so this peptide antibiotic can't bind anymore, or for them to produce their own small molecules with the same shape that bind to the peptide antibiotic so that it cannot bind to something else. Okay, just absorb them all. Okay. So that's an arms race, and there's going to be selection favoring new effective peptide antibiotics, but there's a cost to the ones that are not working or the ones that you are producing even though you don't have the infection. Okay, so for all of these, I want you thinking about their costs and benefits. You could have a much better immune system than you have, but it would come at a cost. And selection has favored different balances of the costs and benefits. Long-lived animals, we tend to have a lot more immune defenses because we are long-lived. Of course, we're limited to what our ancestors evolved. So there's a lot of peptide antibiotics that are really effective against bacteria that attack us that we can't make. We don't have the genes for them. Some other thing invented them. So there have been, frogs, for example, are notoriously good at avoiding lots and lots and lots of bacteria. Granted, there are things that get them, but they can live in water that's loaded with potentially harmful things and not pick up infections that humans will. And there has been some successful attempts to look at frog skin and identify peptide antibiotics that might be useful in humans. Okay, so you'll probably see a question that has to do with, at least one, that has to do with a particular type of invader and what it would encounter on its way to try and infect you, if it went in through the gut or the skin or wherever, and what defenses it might encounter as it was trying to get past them. Could also be a cost-benefit question on these. Okay, any question about those? Okay, just as an aside, just this week, I think it's the second most recent Science magazine, it's been known for a long time, of course, that your intestine has a lot of bacteriophage. These are those viruses that look like little moon landers that have on the tips receptors that bind to something on a bacterium. These are bacteriophage, and they're specific to bacteria. There have been lots of attempts to try and figure out, can we use them to just feed them to people or in other ways to attack bacterial infections, have these viruses, which don't attack eukaryotic cells, use them to control viruses. It's been known for a long time that the mucus in the stomach traps viruses, any viruses, but that includes bacteriophage. So there are images showing that there's about 4 times the concentration of bacteriophage in mucus as there is just in the gut contents, which makes sense. If it's sticky to virus, you'd expect them to accumulate there until it's sloughed off. The interesting thing, though, is that what people have discovered in a cell line, if here's the lumen, that bacteriophage get taken in, they get endocytosed into those intestinal cells. Now that could just be viruses have mechanisms to get into cells, and so maybe they're just piggybacking on a mechanism that gets a virus into cells just by chance. Some of them happen to get in. But the interesting thing about this is that they get moved across to the other side and exocytosed on the other side, which is not what viruses normally do. So this is not like an infectious activity. And now people are wondering, what are they doing? The suggestion is that in all of us, if these rates in these cell lines, which granted they're cancer cell lines, so they're cancerous cell lines, so they're not exactly equivalent, but if the rates that they found make sense, then all of you are moving 30 billion bacteriophage from your gut contents into your extracellular spaces every day. 30 billion bacteriophage. And people are wondering, maybe that's just sampling the gut contents for the immune system to figure out what's in there. So maybe a bacteriophage of a particular bacterium would tell the immune system that bacterium is here, ramp up defenses against that bacterium. Okay. Or it could also be, hey, look, yeah, it takes a bit of space, but these things will attack bacteria that manage to get across the cell barrier in the intestine. So why not load up these intestinal extracellular spaces with bacteriophage as another defense? Okay? And the great thing about this defense, yes, it takes space, but it's no extra resources. The bacteria in the gut are using the resources to make the bacteriophage. So you don't have to have your own resources to do it. So, cool new finding, yet another potential innate immune defense. Okay? And there is still, I would say, far more that we do not know about the immune system than that we do know. Okay, questions? Is this cool? Yeah. Some of you, others going, trapped in this course with Heidemann again. Okay, I want to talk a little bit more about inflammation. I just want to walk you through it quickly. So, got the skin. So here we've got skin outside. And we get a wound, and some bacteria get in. That ought to start a response, right? If you've got a cut, even before you detect any bacteria, you can predict there will be bacteria there. Sterile surgery is not a normal condition in life. Okay, so there will be bacteria there. Just start ramping up the immune system in that local region. Okay, that's what the inflammatory response is. It involves macrophages, but what's happening is you want to get macrophages nearby. Is this to scale, by the way? So here's the macrophage. Here's the bacterium. Here's the receptor. What can you tell me about the scaling here? If this was a bacterial ruler problem, would you be going, you messed up badly? Okay, so how big should the cell be? Bigger or smaller? Bigger, quite a bit bigger. According to the size of that bacterium, this cell is only about maybe 6 microns, 5 microns. It ought to be closer to 10 or 20. So bigger, not all that much bigger, but it would fill the screen. Okay, and maybe even considerably bigger than that. The bacterium would be about 1 to 2 microns is what we'd expect. Okay, what about the size of that receptor protein? Okay, could you even see it if the bacterium was on this scale? No, you wouldn't even see it. Okay, so here's a macrophage. So you want to get macrophages there to attack it. And part of the immune response is that damaged cells, so here's a damaged cell, they release cytokines. There's a whole bunch of different cytokines. Cytokines, generic term for messenger molecules from the immune system. Okay. So, generic term. So, they release cytokines. Those cytokines they increase blood flow. So, where would they be binding? Nearby blood vessels. Okay, so they'd be binding to nearby blood vessels, binding on the precapillary sphincters, right? At the beginning of every capillary, there's a little ring of smooth muscle that'll shut it down or open it up. And it opens or closes depending on how acidic that environment is, which is an indicator of how much CO2, which is an indicator of how much oxygen there is, okay? But when there's cytokines there, they just open up. And same thing with the little arterioles nearby. They relax their circular muscle and they open up. So more blood flow. So more blood and therefore swelling tends to raise the temperature also in that area because with more blood flow you're bringing in warm blood from the core of the body, just gets hotter. So skin gets warmer, probably speeds up the reactions in your cells and your immune system, so that probably helps at least a little. Okay, and you also get, this is part you've already looked at, you get these bacterium binding. When they bind, that activates. So if you had, so if these were myosins and you activated them, those myosin heads would be bending, and that would push this part of the cell out, right? If this myosin head does this, that'll push this part of the cell out. That makes sense? So you grow a pseudopod. Okay? Same molecular toolkit we've been talking about for movement and for amoeboid movement of other things, but you can also use it to grow a pseudopod and send it out around this bacterium. And you can also adjust the cell cytoskeleton to change the shape of the cell. In my head, these little circles, the little rows of circles are actins. I just put a couple of myosins in there, and the zigzag molecule would be tubulins that are forming much of the rest of the cell cytoskeleton. So we change the shape of that cell. So we're adjusting the sizes and the lengths of microtubules, actin. Okay, makes sense? So we engulf this bacterium. What triggers it? Well, binding of a molecule that's on the surface of one major class of bacteria, LPS, lipopolysaccharide, LPS. You don't need to remember the word, although I'd like you to recognize it if you see it, but you can just say bacterial surface molecule. Anyway, it's not on any of your cells, so when a macrophage binds to it, it has to be an invader. And when it binds, it changes the conformation on the inside of the cell, which triggers pseudopod formation. Okay. So the pseudopod engulfs this thing, and you fuse it. You fuse it with a vesicle made by the Golgi that has proton pumps that pump H plus into it. Chloride will follow passively through chloride channels, right? So we're doing, and that takes an ATP. So you can put acid into it pretty easily. Okay? Proton pumps are always pumping H plus, as far as I know, out of the cytoplasm. Could be to the outside of the cell, or they can be pumping H plus into a vesicle. It's still out of the cytoplasm in both cases. Okay? So you make an acid, fuse it with a vesicle that has enzymes, and you digest this thing. Between the acid and the enzymes, you break down that bacterium. Get some nutrients, so happy macrophage, it's being fed. And if it's got foreign molecules, that triggers cytokine release. So now we have two ways to get some cytokine release. One is just damaged cells in the vicinity of that cut are releasing cytokines, inflammatory reaction. If there are bacteria and viruses there, that will sustain the inflammatory reaction. So the damaged cells are going to run out of cytokine. But in the meantime, macrophages that are coming in there can be digesting these invaders, and now they start producing cytokine. So you maintain the inflammatory response. All good stuff. The inflammatory response is really important when you get some kind of a cut, something that penetrates through your skin. What about when you get a sprain or a bruise? First of all, if you get a sprain or a bruise, is the inflammatory response going to happen? Yeah, why? Damaged cells, right? That's what a sprain or a bruise is. You've torn things interiorly or crushed cells or whatever. You're going to have some damaged cells releasing cytokines, so you're going to get an inflammatory response. How useful is that inflammatory response? Okay, in this case, not a good thing most of the time. If you need stem cells to move in and do repair, yeah, they need some space and stuff to work. But for the most part, what's happening is you've got an inflammatory response that is useful if you have invaders. But tell me this, how can your skin, how can the area where a wound takes place, how can it tell the difference between something that penetrated versus something that just crushed or tore? There's no way to do it. Okay, so your cells, those parts of your body have no way to tell the difference between a bruise or a sprain and a cut. So they take the safe way out. They start an inflammatory reaction no matter what. Okay, so take-home message. This is one of those cases where there's really practical stuff from this course. When you get a bruise or a sprain, what should you do? Ice it. When you ice it, what ice does is it causes vasoconstriction, local vasoconstriction. So the inflammatory response is trying to open up all those blood vessels. The ice is causing them to constrict. Okay. And so that vasoconstriction reduces the fluid flow. It reduces the leakiness of those capillaries. It reduces all the swelling. It reduces, on top of that, the rate at which white blood cells can get in there. So normally macrophages would be getting in there, and some of them are hostile. The inflammatory reaction is not perfect against invaders. It also damages self cells. So if you can minimize the numbers that get in there, you minimize damage that's coming just from the inflammatory response. Okay, so icing it, of course, not causing skin damage because you ice too long or too cold, but icing it to minimize blood flow while still keeping cells alive, that's what you want to do. Icing, sprain, or bruise, magical. Okay. And this is the reason. Okay, and you saw the video where I showed a few more steps. Here's the inflammatory cytokine binding to white blood cells. That video, The Inner Life of a Cell, that I hope most of you have seen, maybe all of you, that video, Inner Life of a Cell, that's what it's showing. It's showing a white blood cell, spherical, just kind of happily bouncing around like a basketball in a capillary, and then encountering an inflammatory cytokine. The cytokine causes it to become amoeboid. It activates surface receptors that make it stick to the red blood cell in that location, and then it produces enzymes, metalloproteases, that help to increase, that helps digest the extracellular matrix and lets it get between cells and through the basement membrane and out into the tissues in those areas. Okay. So that's what's actually happening in that particular video. And I'm also showing those endothelial cells getting a little bit more leaky in the process of this. So there they are. Cytokines are expanding the gaps. They're sending more blood flow. Both of those starts giving you an edema, local edema, blood fluid accumulation in that location. And macrophages go there, and as long as there are more bacteria, they keep on producing cytokines, which maintains the inflammatory response, which brings more bacteria in there. You can have so many that you get pus, where you can visibly see all that white gunk, dead macrophages. Macrophages that have ingested so many attackers that they can't function anymore, or they've been starved of nutrients and they're dead. But more keep coming in from the cytokines around the margins. Okay. Okay, questions about that? Okay, this was the easy part of today's lecture, I'm sorry to say. Okay, now we want to talk about the acquired immune system. So the acquired immune system takes a pretty big investment. It takes time to develop, and it takes time to respond. It's not fast. But what it does is allows you to have a huge ramp up in defenses that specifically target whatever invaded you. And you need never have encountered whatever that is before. You can still have a defense against it. So it allows you to attack novel things. The innate immune system mostly enables you to attack things that long selection has favored defenses against those things. Okay. The acquired immune system lets you defend against novel attackers. So this is the overview, but I strongly recommend you guys work through sketches of your own. Trying to remember it from this figure is a nightmare. This is way, way too busy, way too complex. I'm going to break it down some, but I want to go through the basic idea. We're going to start with T cells. I never can decide for sure whether to start with T or B cells, but usually I start with T cells. We're going to start with T cells. And cytotoxic or killer T cells, there's multiple varieties, but they're variations on a theme. So what I'm going to give you is one variation. If you take immunology, really cool course. If you take it with Professor Zwollo, you'll see much more complexity to this system. But this is the basic idea. And it's the same basic idea for killer T cells and for B cells. Let me do an eyes closed exercise here. So what I want to find out is whether you feel like you understand it well enough from the video that we can work some problems as opposed to having me walk you through it, or whether you feel like it would really help me to see it again in another way. Okay, so close your eyes. Close your eyes. Raise your hands if you'd like me to walk you through it and see it again in another way. Okay, hands down. Raise your hands if you feel like, no, I don't need that. Rather work the problems. Okay, hands down. Man, what do you think, Lydia? Half and half or slightly more for walk us through? All right, so I'll go ahead and walk us through it. So Basic idea is you have a gene for a receptor, a surface receptor, which can become an antibody, a gene for a surface receptor, and that receptor is the region of that gene for the binding part, the part that's going to bind to an epitope. An epitope is the part of an antigen, foreign molecule that'll bind to a receptor, one of these immune receptors. Okay? So it's variable in that binding region, which means you can rearrange the gene and have any shape you want. If you think of the binding region as hands, and I could give you any shape, as long as it was roughly this size, I could give you any shape and you could adjust your fingers to match it. Okay? So that's what's happening. You're rearranging that part of the gene. So you start off with this really variable gene, and during development, that gene gets trimmed in these cells down to a subset. Okay, so we've gotten rid of here A1, A3, B2, maybe C1 and C2, and what's left is now a subset in this binding region. And there's enough variation, there's enough different parts that can be rearranged that you have billions of possible shapes and charges. Okay, billions of possible shapes and charges. No matter what foreign molecule gets in, it will bind with some level of affinity to one of these molecules, given how many of these cells you have. Okay, so that then RNA polymerase moves along this thing. You get messenger RNA. The messenger RNA goes through a ribosome, and you get a receptor that has a variable binding region. So this would be A2, B1, C3 is that region. Okay, that makes sense? Okay, so if you had 1 billion cells, in principle, you could have 1 billion shapes. Yeah, you get some repeats, okay? But in principle, 1 billion shapes. Okay, and that's the order of magnitude we're talking about for your immune system. Okay, then these things are just going around in small numbers, okay, not doing anything unless something binds. If something binds, that activates a second messenger cascade. So let's jump ahead to that. Okay, so here we have these unactivated cytotoxic T cells that differ in these binding regions because the sequences for those tips are different, okay? And you've got millions or billions of these. Okay, what might they bind to? Well, those receptors, they match MHC class 1. So MHC class 1, another highly variable molecule, and all healthy, all body cells produce MHC class 1. It's another immune system molecule. It does two things. One is, part of it is variable and it's self or non-self. So this one, you all have, unless you have a twin, identical twin, you all have different MHC class 1, but every cell in your body has the same MHC class 1. Okay. These are not variable between cells in your body. These tell you that, if you think about it, all of us are getting bits of live cells from other people all the time, especially if you ever kiss anybody. Okay. If you ever kiss anybody, you're sharing a few cells that are not completely dead and that could just attach to you and start growing, and you'd be a little part of somebody else. Okay. But they have different MHC class 1, and macrophages that bind to an MHC class 1 that isn't self, they kill it. Okay? So a kiss is actually beginning a very hostile series of events. Yes, Ms. Spencer. Talk to your neighbor for a minute about what we just talked about while I think about it. Okay, can you hear me? Somebody very helpfully took dead batteries and put them back in the box so that when I changed them the first time, I put dead batteries in to replace dead batteries. Professors, not always the most sensible people. You can tell that to other professors if you want. Okay, so hostile series of events. Okay, so part of what MHC class 1 is doing is labeling cells self or not. Okay? The other part of what they're doing is that a part of that cell is like a little folded hand. It actually is shaped more or less like this. It has a alpha helix, and a beta plated sheet in between them. Okay? And so it's like a little hand folded like this. And in the Golgi, when these things are being made and prepared, fragments of peptides that are just in the body, short sequences, I don't know, 10 amino acids or so, get put in there. Okay? And so these MHC class 1 molecules are then moved out to the surface, and they have, in a healthy body cell, only self peptides. And for a reason we'll come back to, that means your immune system cells won't bind to them. They ignore them. Okay. So this says self cell, healthy. Only have inside me what I should have inside me. So that's a healthy. What about a bacterially infected or virus-infected body cell? So here are proteins from virus or bacterium. Some bacteria penetrate cells and infect them. Some live outside cells and attack things from the outside. And peptide fragments. Okay, so what we're looking at is when this thing is made, we put in both self and non-self, but here's a viral peptide, or bacterial. And we put it in here. I represented it by the half plus. And then that gets moved out to the surface of the cell. Okay, so what we have is infected cells are putting this message out there saying, infected, I'm infected, I'm infected, I'm infected. Okay? Yeah.
At the beginning of infection, would it take a certain threshold to be able, would some MHC1 present self and some have non-self?
So early on, it takes a while to ramp this up. We don't actually have a lot of good data on this, but there are some theory papers that speculate on it. Once the thing is just beginning, you'd have a lot of cells that just never happen to encounter a other cell, a potential T cell, well, a T cell that has a binding protein to match. And individual cells won't have very many of them, and so even if the right T cell comes by, it's still not going to encounter an actual foreign foreign peptide. Okay, so it takes a while to wrap this up, and there's an element of probability in this. So one of the reasons why one person might have a really severe infection and the other person not could be because the one person, just by good luck, happened to get immune system cells there quickly, and the other person, by bad luck, they just didn't get there. And so the infection had an extra day or two to get going before the body started responding. Okay? There's an element of luck as to whether or not you survive an infection or not. Okay? Yeah? Lauren, right? No. Oh, Claire, yeah, go ahead.
Are there viruses or bacteria that mimic self-cells?
Are there viruses or bacteria that mimic self-cells? I've never read of any that basically that can do an MHC1, and they pretty much need to have surface receptors and molecules that are not self-cells in order to do what they do. Partial mimics, yeah, I couldn't tell you. Professor Zuolo would be a better person to ask for that. Partial mimics, and certainly ones that fool the immune system, absolutely. There are ones that fool the immune system and that produce peptides, for example, that block cytokine release and things like that. Yeah?
Are there any bacteria or viruses that prevent the production of those MHC receptors?
So are there any that prevent production? I've never read of any, but I am not an immunologist. And if we can think of ways to screw this system up that are conceivable for bacteria and viruses, there's a decent chance that some do it. So it's a very reasonable hypothesis, but I don't know whether anyone's actually tested it. So both of these actually are reasonable hypotheses, and I don't know for sure whether they've tested them. Okay? Okay, so what happens? All right, so second thing that happens is that fragments of bacteria or virus also wind up on what are called antigen-presenting cells. So these are immune cells who have as one of their functions, they put fragments in another MHC, but this is MHC class 2. So MHC class 2 is only on immune cells. Okay? So MHC class 1 is a way for all body cells to say, well, except for immune cells, all body cells except for those to say, I'm infected, I'm infected, I'm infected, or I'm not, and also to say, I'm a self-cell. Okay? MHC class 2 is a way for immune system cells to say, look what I found. I got this thing. Okay? I found this infected thing. Let's do something about it. Okay? So MHC class 2 got this thing. Okay, so now we have, let's go back. We have infected cells with this molecule, this peptide on the surface, and we also have immune system cells in MHC class 2. So we have MHC class 1 and MHC class 2 that have this small particle from a bacterium or a virus. Okay, so what happens? We can do these in either order, but in this is the antigen-presenting cell. Okay, so that's an MHC2. And if one category of T cells, helper Ts, I wish they weren't called T cells. So when T cells were first found, then people started finding subcategories and they wound up with names. I wish these were just named helper cells or were abbreviated H cells all the time. But anyway, helper T cells, so it's a different category of cell. Their job is not to do anything directly. Their job is to provide an additional signal. Okay, so when helper T cells, if they have a receptor that binds, and by the way, also variable, just like killer T, which we're about to talk about, and B cells. Okay, so only some helper Ts are going to be able to bind this. Okay? But if they do, then they release cytokines. Okay, so if they bind, And of course, that's only going to happen if it's a non-self molecule there. We're going to come back again to why in a moment. So that's one message of an invader. The other message of an invader is, here's our unactivated killer T cell. And if its receptor binds, okay, so that receptor can bind to an MHC class 1 if there's the right peptide in there, it gets activated. So if that peptide fits, so if that peptide, this thing, if that fits the receptor here, okay, then it can bind, and it'll activate, change the conformation in here, activating a second messenger pathway. And if it also gets this cytokine, so remember this was our helper T, okay, it's our helper T, it had to bind to an MHC class 2 over here. So if MHC class 2, two messages. To activate our killer T cell, it needs to both bind an infected cell and get this cytokine. Okay? And that cytokine is only going to be abundant if this happens near an antigen-presenting cell. If that happens, then we get a pair of second messenger cascades. They both have to be there. And we get cell division. Many rounds of cell division. Why two messages? Why not one? The idea is that if you just respond to this infected cell, the chance of false alarms, the chance of errors, the chance of autoimmune disease is much higher. If you require two messages, it's a little safer. Autoimmune diseases are dangerous things. They're way too common. Okay, and having that requirement for a second message probably reduces the chance of an autoimmune response. Okay. Anyway, if you get both those things, you get many rounds of cell division. So here's our cell division. And you wind up with many copies of mature killer Ts. These are no longer naive. They all have the same A3B1C2, A3B1C2. They all have the same sequence, so they all have the same shape of their receptor. But now they're mature. So if they bind an infected body cell, that activates a second messenger pathway. And that second messenger pathway causes release from vesicles, things like enzymes, perforins, also causes the production of reactive oxygen molecules, H2O2, hydrogen peroxide, and others. They're called reactive oxygen species. Terrible choice of terminology when you're in a biology class. It's fine if you're in a chemistry class to talk about reactive oxygen species, because everybody knows you're not really talking about species. But talking about them in a biology class, we should not be forced to use their terminology. Anyway, reactive oxygen species, in other words, things like hydrogen peroxide, that have these highly reactive forms of oxygen, release them on or right next to this infected body cell and you kill it. And you also destroy everything inside you. So macrophages come in and ingest everything inside. So you destroy these things. Yeah, Katie. So the question is, are they released directly into the infected body cell, which would be more effective in targeting just that cell, or nearby? I've not happened to find out. So I've looked at some reviews and they haven't happened to tell me. Jake?
So the later killer T cells, they do not require helper T cells?
They no longer do. They're now mature. So once they're activated, they just need to bind to an MHC class 1 with that antigen on it, which identifies that cell. Okay? So now you've got all these things destroying the cells, and you have macrophages coming up and ingesting the bits. In addition, others, after division, stay inactive. So some small subset stays inactive. They are memory T cells. So if you get a new infection, you've got thousands, tens of thousands, hundreds of thousands of these. If you get a new infection, they can attack right away. They can also be prompted to go through cell division. This whole response is faster. Okay, on Friday, we'll go faster through the same thing for B cells, talk a bit about why these don't just attack all your cells, and we'll do some practice problems for the final. Okay. If you're going to celebrate wildly the last day of classes, like, say, the Thursday night before, and you're not in good shape for class, just don't come to class. Please don't disturb anybody's class, okay? I'd much rather have you here alert and aware.
Are the cytokines that